This program is made possible by a grant from the National Science Foundation, a co-production by Mississippi Public Broadcasting and the University of Southern Mississippi Gulf Coast Research Laboratory. April 20th, 2010, 41 miles offshore in the Gulf of Mexico, BP's Deepwater Horizon drilling unit exploded after high-pressure methane gas engulfed the rig and exploded. 11 people would lose their lives that night. For nearly 3 months, oil gushed into the Gulf of Mexico. Marine life, tourism, and petroleum operations will never be the same in the Gulf region. Now there are questions that have scientists focusing on trying to shed light on the unknown long-term effects on the Gulf. This is the Science of the Spill.
Hello and welcome to the Science of the Spill. I'm Erin Pickens.
And I'm Dr. Bob Thomas, Director of the Center for Environmental Communication at Loyola University.
Well, the Science of the Spill is a three-part series produced by the University of Southern Mississippi Gulf Coast Research Laboratory and the Mississippi Public Broadcasting to inform the public with scientifically proven facts concerning the BP Deepwater Horizon oil spill. And Dr. Thomas, I think we can all agree that there are many myths and information out there that are not based on scientific data about the effects of the spill.
Well, that's right, Erin. And we're going to bring together scientists that are studying this disaster for discussion to give the facts about issues related to the Deepwater Horizon oil spill. In today's program, we'll be discussing the impact of dispersants on the environment and also the overall health of the marine life in the Gulf.
Now, we will also let you know that you can find more on our companion website at SpillScience.com. There you can find in-depth information about topics related to the spill, as well as video discussions from scientists studying the effects of the spill on the Gulf of Mexico. You may also want to join the discussion on our blog section of the site, where you can engage in discussions with scientists studying the impacts from the Deepwater Horizon oil spill. Again, that's SpillScience.com. Now, during the early stages of the spill, a large amount of dispersants were dropped to help break up oil found on the top of the water. Southern Miss scientists at the Gulf Coast Research Lab and CENIS have posed many questions about the impacts of dispersant agents often used to respond to oil spills.
The dispersant issue is a very interesting one. The idea of putting the dispersants in with the oil is in principle a good idea. You'd like to widely disperse the oil to make it more available for microbial degradation. That's the basic concept. Now, the problem, of course, is that you're adding another chemical to the mix.
The main dispersant that has been used is a compound made by a company, Nalco, and it's their Corexit 9500. There are 6 ingredients in Corexit 9500s. All 6 of them have been approved for various uses in human products, ranging from toothpaste to pharmaceuticals to propylene glycol as antifreeze in your radiator. And propylene glycol is probably one of the most toxic compounds, and it's not terribly toxic. So the role of these dispersants is to work as a surfactive agent. And a surfactive agent is nothing more than a detergent. One end of the molecule likes oil, the other end of the molecule likes water, and so it physically pulls the oil apart into very small pieces that the microbes can then interact with.
One thing that's kind of interesting in those numbers that I really haven't seen much public comment on is they say that 8% of the oil was chemically dispersed. Now, of course, these dispersants are things that people have been very concerned about. So a question to be asked is, first of all, how good a number is that 8%? And if that's it, then from a policy perspective, does it make sense then to use the dispersants if that's all that it's affecting?
The controversy is that some scientists believe that the dispersant is toxic and that we're underestimating the toxic effects. Other scientists believe that it's not terribly toxic, but like all chemicals, if you apply enough of it, it can be toxic in large quantities.
EPA has a certain set of tests that it runs, mainly on one type of organism looking at toxicity. Some people have suggested that maybe there need to be toxicological studies on different sorts of organisms and perhaps for longer periods of time. And in fact, some of the research that the National Science Foundation has funded for this oil spill is indeed looking at some of those questions.
The one thing that I have done in my laboratory is to look at the decomposition of Corexit. And in my laboratory, in what we call microcosms, simulated replicas of the environment, we've simply come down here to this dock, collected water from this bayou, and we know full well that there are oil-degrading microbes in this water, and we've used that to take a look in my lab at the decomposition of oil as well as the decomposition of Corexit. As scientists look at the fate and effect of dispersant in salt marshes, in marine animals, we'll have a much better handle on what the effect of the dispersant is as a result of the Deepwater Horizon incident than we did before.
Now, we've heard some important questions raised by Dr. Schiller and Dr. Grimes about the use and effects of dispersants. We have Dr. Thomas now here with scientists that can help answer some of those questions. Dr. Thomas.
Well, thank you, Aaron. I'm here today with Dr. Joe Griffith, who's an assistant professor in the Department of Coastal Studies at the Gulf Coast Research Lab, and Dr. Christine Willett, who is a professor in pharmacology and environmental toxicology at the University of Mississippi. Welcome. Good to have you with us. If we could start today by talking a little bit about your research program, and then we'll get into some specifics about how that affects the work on the Gulf. Okay. Christine?
So I study the mechanisms that a particular class of chemicals that's in oil, the polycyclic aromatic hydrocarbons, and we typically just call them PAHs. And I study how they cause their toxicity. And so we've been using primarily fish models, and we like to look at these chemicals are known carcinogens, and so we study about how they cause cancer, but then we're also moving now into how they might cause this thing called endocrine disruption, how they might interfere with hormones, and also maybe reproductive effects. And so as we move into the oil, then we're thinking about whether the oil could potentially be causing some of these same things. In addition to that, we have an analytical aspect of our research, wherein we just collect samples along the coast, like water and sediment, for example, and measure the concentrations of PAHs in those. We actually started this project right after Hurricane Katrina hit, so we sort of saw the impact of Hurricane Katrina, and now those levels have returned to baseline, and so now we're poised to see the impact of the Deepwater Horizon spill at those same sites. And those sites were from Gulfport to Mobile, so we have a nice chunk of the Mississippi coastline to review.
Well, it's comforting to know that you have a baseline of information you can go back and compare it to now. Joe?
Yeah, so as soon as the incident happened, my lab started focusing on two different lines of research. One, we went on the boat 3 or 4 days after the Deepwater Horizon rig collapsed, and we were sampling in the Mississippi Sound and along the coastline of southern Mississippi for species to establish our own baseline. So we collected a large number of samples. I think the last time I checked, we've got 970 samples sitting in the freezer of different species, finfish, shellfish, oysters, things like this, so that we would be able to go back to the same sites later on and establish exactly what the effect was by examining the differences between the two. So we've got a large number of species in the freezer ready to be analyzed. Simultaneously with that, we're doing some laboratory-based studies. We've obtained crude oil and dispersant from BP, and we're mixing that together to produce what we call dispersed oil, essentially the mixture of oil and Corexit that we hypothesize the animals are being exposed to in the Gulf. And we're exposing different species to that under very carefully controlled laboratory conditions. The idea being that in the laboratory we can isolate out all the different environmental effects that might be confounding the response and isolate and identify just exactly what the effect of the dispersed oil on different organisms is. And we have 3 different exposure chambers in the toxicology building at the Gulf Coast Research Lab, and we've got all 3 of them busy constantly. We have one that just started on Friday. We're looking at chronic effects of dispersed oil exposure to a small fish that's indigenous to the region called the sheepshead minnow. We're simultaneously running exposures to some of the shrimp species that are indigenous to the area. We grow those in culture at the Gulf Coast Research Lab, and so we're doing exposures both on adults and juvenile examples of those species. And we have, along with my colleagues, a NSF Rapid grant to look at the effects on blue crab larvae. So we're exposing those guys to crude and dispersed oil to identify the effects on them as well.
Well, that's excellent. From the video, I think it was pretty apparent that there are some bona fide reasons for using dispersants when you have an oil spill. I'd like each of you to comment on it in your labs. What are you seeing about the residual toxicity of the dispersants and the breakdown components?
Well, I'm really not going to talk too much about what we've seen. It's still very, very preliminary, and we want to make sure that we have it vetted through peer review to make sure that that our results are valid and that they're agreed with. We're much more concerned with being accurate than we are with being right on this issue. I'm sorry, we're more concerned with being accurate than with being quick on this issue. I understand that people want to understand, want to know what's going on, but nobody's served if we have information in the press or in the public that later turns out to be inaccurate. So we're going to wait until we have our results reviewed by other scientists, and then we'll release it.
Good. Well, would you, and you kind of answered my next question right there at the end, but before we go to Dr. Willett, tell the audience what you mean by peer review and why that's so important to a scientist.
Sure. So peer review is essentially the way that we try our best to make sure that no inaccurate information gets published. And it's not perfect, no process ever is. But essentially we'll complete a section of research, we'll come to a conclusion about the effects, and we'll write it in a paper for publication. And we'll send it to a journal, and the journal will then send it out to other scientists entirely unconnected to this. I don't know who the reviewers are, and they'll read through the paper very, very carefully, and then they'll try to tear it apart. They'll try to find every weak point in it, and then I'll get a version of it back in the journal. I say, well, it's rejected or it's accepted, but you have to address these comments. And it's a series of very rigorous, very focused comments on exactly what is this, why did you do this, how do you explain this in light of this, and then I have to write a response to that addresses each of those concerns, so that by the time it's been out and it's actually published, it's been through one, maybe two rounds of review where other people who aren't invested in this research are making sure that what I'm saying is accurate.
Well said.
Sure.
Dr. Willett.
Yes. So the question is back to what the dispersants. So we haven't been addressing that exactly, but what I can say is that the potential from the dispersants is now that the chemicals that are in the oil are now dispersed through the water column. So if you've got the surface slick on the top, then organisms that have to come up through that slick, your sea turtles, your dolphins and things, are getting exposed to that slick. But potentially things under that may or may not have an impact. Well, once you disperse that oil, things like seagrass habitats or oyster reefs or so forth could now be, those oil constituents could now be bioavailable. So some of the work we're doing is we've collected, right as the spill happened, we collected seagrasses and oysters, and we've been collecting those throughout this time course. And what we're going to try and do is see so those ones before the spill are our sort of baseline, as you might say. But then we predict there might be some sort of stress response in those organisms to oil or dispersant or whatever was there. And then we're going to look for a return to that original condition or recovery. And we're doing this by what's called proteomics. And so we're looking for a suite of different proteins that are expressed in those organisms to see if we can, sort of track this change, track the stress, and then perhaps the recovery. So again, these organisms might not have been exposed if the oil had stayed on the surface, but potentially now we might have impacts in those kinds of organisms that live, below the water line.
Okay, now let's stay on your lab's work for a moment on the next question. And that is that, how do you express toxicity in these animals? I mean, how do we know that something is toxic to a group of animals?
Right. Well, the key thing in science is to have controls. You need both positive and negative controls. And so in our case, the baseline or the before the spill are what we're comparing back to. Or if we're bringing in water, say we're doing a bioassay in the lab, we do embryo exposures in fish, and we're taking water in real time from the coast and comparing it. So then we'll have just a laboratory water that we're comparing those controls to. So you're basically saying it's the percent affected compared to controls. And then for a positive control, you can use just pure oil or constituents that are in that oil where you really know what the effect should be, so that you can sort of see where in that continuum your organisms fall.
Anything to add to that?
Well, I guess the one thing that I would like to add is that it's not really accurate to say is something toxic or is it not toxic. Toxicity is entirely a relative term. You know, we have a saying that Paracelsus's law, you know, the dose makes the poison. Anything is toxic at a high enough concentration, even water. And at a low enough concentration, there's nothing that can cause toxicity. So the toxicity that we'll see to different organisms will and is depending entirely on how much oil they're exposed to, dispersed oil, and how long they're exposed to it for, as long as, you know, different life stages, things like that. But it's the amount of dose that they get and the time they're exposed to it that's really going to be the prime determinant of the effects that we see.
Well, are you looking at mostly the toxic effects on individuals or on populations or subpopulations?
Well, so, you know, initially we'll start out by looking at individuals. Initially we'll do what we call a kill 'em and count 'em test. We'll try to establish what concentration is required to cause overt mortality in our species. And that's not really because we expect that concentration will be reached in the environment, just that it will then guide us in picking other concentrations that might be more relevant, because we don't have very good data as of yet on actual concentrations in the water that the animals are actually living in. So we'll do that first, and then we'll do a series of other assays. We'll look at individual responses, but we always try very hard to tie it into an organismal or ecosystem level response. In other words, we'll look at reproduction effects. Are the animals reproducing? Are the juvenile forms growing? Are they going to grow up to be able to be reproductive? You know, are the eggs hatching or are they not? And it's those sort of endpoints that we then try to tie all of the individual level responses, use that into the organism or ecosystem level.
Right. I think that's what a lot of people's questions are, is what's going to happen to next year's productive stock. You know, are we going to have, because we have crabs today, we have shrimp today, are we going to have them next year?
Sure.
So people are very concerned about that. Well, what, you know, we're talking about dispersants, but what about the toxic qualities of oil? Either one of you?
Sure. Well, that's what we've been studying prior to this, and the important thing to remember is oil in itself is a complex mixture, and the oil that came out on day one very quickly starts changing. There's some very light or small molecules in there that volatilize very quickly. That while they can be acutely toxic, they're not around for very long. And then what happens is the oil becomes what we call weathered, and then the higher molecular weight, more persistent molecules remain. And so when you're studying oil toxicity, it's kind of tricky because you have to really define what the mixture is that you have. And one of the things we do is pull out individual chemicals to really, again, narrow it down so that we can try and figure out what the mechanisms are. So I think one of the things we do know about oil is it is can be acutely toxic, but then we study fish embryos, and the nice thing about fish embryos is you can watch them develop, right? They're not internal, they're not inside the mother, and we can see one of the big things we see from oil or PAH toxicity is cardiac defects, and so that's a heart, so that their hearts aren't forming properly and they're not beating, and that's obviously going to affect whether they can hatch and then survive. Other concerns, like Joe said, is reproduction, and that's a little bit trickier to measure because, you know, it's a complicated environment. Even in the laboratory, it's sometimes complicated to measure. But the impact could be on 3 generations out. So it could be your grandkids in relation to the exposure. And so that's, again, something we're trying to study in the laboratory with very controlled experiments. But you know, it takes multiple generations to see that, so it's going to take some time. But that's one of the things we think that the constituents, at least in oil, could be doing.
Right. Yeah, I think there's a lot of concern from the Valdez spill about the 4-year lag before the herring fishery dropped, and I think that's got people very concerned down here because people understand it takes a while for science to produce solid results that we feel comfortable with. but everybody's wondering down the road what's going to happen to the fisheries out there.
Sure.
Well,
Any
we're
comments?
going to be monitoring it. I mean, we're going to be
Right.
actively looking at it, and given the available resources, we will be monitoring this on a long-term basis. The Gulf Coast Research Lab, you hear from some of our other people later, have a very long-term database of abundance of different species of fish and shrimp in the Mississippi Sound and surrounding waters, and so we'll be able to say with a high degree of accuracy, I think, what the effect is on the organismal or ecological level based on comparison to that. And we'll, from my lab, we'll hope to add in trying to understand what changes at the individual level are driving that response.
Exactly. I think those are questions everybody's looking for answers for. So now we've introduced talking a little bit about dispersants. We've talked a little bit about oil. What about the two together? That always has everybody concerned. Where do we go from there? Is anybody working on that? And if not, when do we get there?
Well, that's what we're actually. I actually brought Christy a sample of the dispersed oil that we made in my laboratory. We got, we mix the two under precise conditions, and we make what we call dispersed oil, or the water accommodated fraction. And it's that we're actually using in our exposures in the laboratory, because it's unlikely, I think, that anything inshore is going to be exposed to either pure oil or pure dispersant. It's more likely the mixture of the two. The dispersed oil, that's going to be, like you said, much more widely distributed throughout the water column than just the crude oil on the surface of the water is. So that's what we're using in the exposure, and that's what we'll be exposing dilutions of the WAF to different organisms for different lengths of time to look at very precisely what the effects are going to be.
Right.
Yeah.
Anything to add?
Not necessarily, except for that, again, and I think Joe's going to do this, is we have to look at not just whether it's going to kill them, what are sort of the different sensitivities, the different life stages that are important in different organisms. Everybody has a different sensitivity, or every creature has a different sensitivity to these things. Yeah. And so that's some of the work he's addressing.
Yeah, absolutely. That's what we'll all be doing. And I think one other thing to mention is that, it's very, very difficult to extrapolate across species. Sometimes we have to do it, but not all species, even all fish, respond the same way. There can be very big differences in sensitivity, and so we can't test every species in the water. We don't have the time, we don't have the resources, the manpower to do it. So we'll try to pick some species that are representative and extrapolate out. But we have to admit there is some inherent uncertainty in doing that.
Right. Well, in a 2-minute summary of what's happening in your lab. Go back to the high points.
The high points. Well, I think one of the key issues as we go forward is we need the continued funding to keep looking at this, because it could be a couple generations out. And so I don't want this to be sort of a flash in the pan, and really use this, we're in a university setting, to use this as an educational opportunity to get at, so this doesn't happen again, or if it does happen again, how do we do it better? How do we clean up better? How do we protect the organisms better? And so that's what we need, this sort of recovery, tracking the recovery, but also getting at, how can we use dispersants again? Is there a better way? And so that's what I think all this research is going to help us answer.
Excellent. Dr. Griffiths?
Yeah, I think Christy really hit it. One of the primary concerns is going to be continued funding for research. This stuff is not cheap. Invitrogen doesn't charge me less when I go and ask them, buy a reagent because there's an oil spill out there, and we have to have certain expensive reagents to do the research and to do it right. And so continued funding that allows us to look at multi-generational effects, at ecosystem level effects, at multiple species is going to be absolutely crucial, and we're hopeful that materializes, but we'll have to wait and see.
Well, excellent. Thank you for being with us.
Thank you.
Aaron?
Thank you.
All right, thank you so much, Dr. Thomas. Now, one thing we all think of when we hear about the Deepwater Horizon oil spill is the impact on marine life in the Gulf. Scientists from the University of Southern Mississippi working at the Gulf Coast Research Laboratory in Ocean Springs and the Stennis Space Center in Hancock County have been studying the effects of the oil spill in life in and near the Gulf of Mexico. We talk with some of these scientists and researchers on the coast.
Oh, I think there are a number of issues, including the seafood safety issue, which is being addressed by FDA, by our DEQ, state DEQ. Those people are conducting a series of rather extensive tests to determine the safety of the seafood here along the Mississippi coast.
I think it's critical that our agencies who are charged with those functions continue to do that, so that they give people that reassurance and I hope that that monitoring will continue. The University of Georgia just came out with a study which is almost the opposite of the previous findings, in that they feel that most of the oil is still in the Gulf. And so that is a question. And I think it's important to continue to look. We need to look for answers. We need to understand what's happening to the Gulf, even above the seafood safety issue.
Other sorts of scientific questions are more of the long-term questions. What's going to happen to the fisheries down the road? Certainly you can see in the news what happens when, say, a bird or a turtle gets oiled, but possibly the big damage is on the larvae of the fish and other organisms, and it's very difficult right now to see what has gone on, how much has gone on, and we're really only going to learn the results of that exposure 1, 2, 3, 4 years out, when we see, do the fish come back?
We've reached a point now where 4 months out, from the original spill, they've gotten the well capped. We're starting to look at longer term, larger scale sorts of impacts. And that has to do with, are we going to see an impact over the next few years in the incidence of disease in fish? Are we going to see impacts over the next few years in reductions in growth or reductions in population size that continue? When you start talking about longer-lived species, like many of the fishes that live along the coast, then we may not see the results of the impact for several years.
Actually, the state of Mississippi is very fortunate in that the laboratory has maintained a 37-year database on abundance of juveniles of our estuarine-dependent species. So we have a lot of information that we can use to assess change.
I would like to know, what is the oil doing in the ecosystem? What's it doing in the salt marsh? What's it doing to juvenile fishes? What's it doing to different kinds of larvae, crab larvae, oyster spat, all the various things that grow in salt marshes and in shallow waters.
Biologically, it's imperative that we understand just what happened. And again, that may take time. But the scientific process ultimately, I think, will provide the answers to many of those critical questions that both scientists and the public, our residents, our people, all of us would like to know.
And we all know that seafood safety is an important issue to the consuming public, but scientists at Southern Miss are looking at long-term considerations that impact the overall health of the Gulf environment. Let's turn our scientists here today to discuss more about that impact. Dr. Thomas.
Good. Thank you, Aaron. We're happy to welcome today Harriet Perry, who's the director of the Fisheries Center at the Gulf Coast Research Lab, and Jim Franks, who's a fisheries biologist and a senior research scientist at GCRL as well. Good to have you with us.
Thank you.
Nice. Let's begin. Harriet, if you would give us a little overview of your lab's work.
I work in fisheries, and one of the things, of course, we're concerned about with the spill is the impact on distribution and abundance of commercially important and ecologically important species. And we have monitored for the past 37 plus years, and we really stepped up our monitoring efforts when the spill happened. And it will take us probably a year before we can really evaluate the impact of the spill on distribution and abundance, because different organisms have different seasonalities, but we feel very confident that within a year that we will be able to look at changes and to assess the impacts.
Jim.
Yes, well, of course, Harriet and I have worked together for many years at the laboratory, and she mentioned species of fishes, invertebrates of commercial value. Of course, many of those are also of recreational value. You have a very important, a very large recreational fishing industry on the coast. It has been impacted by the spill, of course, in many ways. A lot of those species, those fish, are extremely important in that industry. and we have great concerns for those as well. Various life history stages of those, both in the estuary and offshore, are of very high interest to us at this point, and we are in the process of conducting a number of studies to examine potential impacts to those fishery resources.
Mm-hmm. Well, I think it's important for the audience to understand that y'all have been at this a long time. You said that, you know, your labs have tons and tons of data, a good baseline to spring forward with. Based on that information, and now looking ahead at this oil spill and what it's done, what's happened in the Gulf of Mexico, what are you anticipating? What are you thinking may happen to, what are your concerns about what may be happening to the fisheries, and is there anything that you've seen so far?
I think our initial concern when the oil spill first hit it was such a horrific environmental catastrophe. Are we going to see fishery failure? Are we actually going to experience what other estuaries and coastal ecosystems have experienced? Or will there be no effect, or at best, if there is going to be an effect, will it just be a diminished year class? And as we have sampled over the 4 months or 5 months since the spill, it appears to us that we're going to probably be looking at perhaps a diminished year class type of scenario as opposed to any kind of catastrophic fishery failure. And that's very encouraging. Mm-hmm. One of the things that's important to understand about Mississippi fisheries is that most of them are estuarine dependent, which means that they spend their larval life history offshore, and they use our coastal wetlands to grow and to mature. And even if oil had never hit Mississippi, it would still have been possible to have an impact on our fisheries because of the offshore larval stage of many of our important fishes and invertebrates. So when the spill happened, those animals and organisms that spawn in the spring, their larvae were offshore. And we estimate about 40% of the larval grounds were covered with oil. And so I am currently monitoring blue crabs very heavily, because their larvae were offshore, and they will continue to be offshore until probably October when they cease spawning. So I will have accumulated probably almost 2,500 samples that we will be going through this year just on blue crab settlement.
Hmm. As a follow-up to that, of course, shortly after the spill, I was involved in a research cruise offshore. We were focusing on the young of the big offshore species, the tunas, and some of the billfishes. We were able to collect a number of samples in the southern Gulf and up in the northern Gulf within an area that had been impacted by the spill. In fact, we collected some of our samples under the oil sheen. And so we're actually processing those samples now just to, first of all, to see what's there. Particularly interested in the giant bluefin tuna larvae. This is a very special fish that spawns in the Gulf of Mexico. Its peak spawning season is April and May, and historically our samples have come from the region near where the spill occurred. So there's great concern for that species, and so our sample analyses continue to see what we actually have found in our rather large number of samples. That's an ongoing process. And I did bring back some of those samples, of course, for our toxicologist. Hopefully someday he'll be able to examine those as well from a tox point of view to see maybe what some of those impacts from the spill had upon those species. So both inshore and offshore, there's this linkage. You know, the offshore spawning, the early life stages, later on many of those species coming into our estuary and actually form the basis of our seafood industry. But in the case of those large pelagic fishes, those fish do remain offshore. And although the adults might have been able to avoid the spill, once they spawn, their eggs and larvae are passively drifting at the surface in currents, and we were concerned that a number of those may have been transported into the spilled area. So those are quite important concerns, I think, for us at this point.
Yeah. Well, you know, y'all have made the point that there are just so many elements of the biology of these critters that you have to study, and you have to gather information on the impact of the dispersants in the oil. You've got decades of information out there. I'm interested in two things. Do you just keep doing the same things but start to apply it to the effects of the oil spill? And at what point do you think you can start to come out with information on what kind of a timeline?
For my group, we're actually ramping up sampling. We want to do a better job of sampling the early life history stages, and that would be the stage that moves from offshore inshore. So we use a variety of different kinds of gear depending on the size of the animal that we're after. So for those early life history stages, it's very time consuming, and it's very time consuming to pick the samples. And so we have indicator stations, but we hope with funding to be able to really spatially broaden our sampling area and the number of samples that we can do so that we do adequately address settlement issues.
Yeah, I think Harriet and I are in the same boat on that one, no pun intended. But we do intend to go offshore and to do some advanced studies inshore, and hopefully in the next week or two, to go back in those areas where we had previously sampled before the spill, and some of those samples were taken during the spill. To go back and look at those areas, get some new samples, and to again assess those and see if we can determine some impacts based on what's happening right now.
Yeah. Are there any preliminary results you could discuss? And it's okay to qualify it, but please explain to the audience why you would qualify it.
Could I add a little something to what Jim
Please.
said that I think is an exciting area of research that the laboratory has conducted. In past years, we will be taking a cruise to look at the deep water ecosystem of the deep sea crabs, and our laboratory along with the University of South Florida and the University of Florida in the 1980s and 90s did an extensive study, and one of our study sites was very close to the wellhead, and so we're going to revisit those sites, and this is very deep water, about 450 to 500 fathoms, and we will be retrapping to look at impacts of the spill on those deep water organisms.
And on that same cruise, I'm going to be, along with our team, our large pelagics team, examining the composition, the species richness, the numbers of species, the distribution and abundance of fishes and invertebrates that reside in a drifting habitat known as sargassum. It's a pretty spectacular surface habitat. It's a form of algae that's a spectacular plant that covers vast areas of the Gulf, and is really one of the major habitats for the early life stages of many of our offshore species. And of course we have encountered that in quite heavily oil conditions over the last few months, and we're concerned about the impact and the insult to that specific habitat and its inhabitants. So we'll be examining that as well. And many of those fishes are those young fish later grow up to become the base of a number of our commercial and recreational fishes. So we're very interested in assessing those impacts as best we can.
Yeah. Harriet, I remember a preliminary interview that you did where you talked about that y'all had found the evidence of oil in the larval blue crabs, and the media picked that up and ran with it, and I think maybe went a bit far, the scientific community would say. What did that mean to you, and you countered that?
Somehow it got wrapped up in the seafood safety issue, and there is a difference in being fit for consumption and being biologically fit. And while a fish, the flesh may be fine to eat, and all of the tests, whether they're federal or state tests, have come back that the seafood is safe, is not the same as saying an organism is biologically fit, in that there may be, from chronic exposure, you might see changes in their reproductive capacity or changes in growth, and those are important fishery issues. We certainly don't want to be selecting for small size and have reproduction curtailed. So I think it's important that the public understand the difference in those two. And we were concerned with, from an ecological standpoint, in finding those small droplets. And we are still working on that research with several other laboratories to try to understand what we are seeing. And it's going to take some real funding to help us with analytical cost. We've been kind of doing it on a shoestring budget, but we are continuing to monitor and to send samples out for analysis.
Right. Yeah, that complicates discussion in the lab, doesn't it? On that same article, I was interviewed and I talked about bioaccumulation, but I was talking about in the whole ecosystem of how this oil can move up, and people took that to mean that all of a sudden adult crabs would be full of oil.
Right.
And we didn't mean that at all, so that's a real concern. But that leads to the next question, and I'll address Harriet here, but Jim, if you jump in. There's been a little controversy here about how much oil is left in the Gulf. You know, we heard NOAA make a pronouncement, and a lot of us think that was misconstrued. It sounded like that only 26% was left, whereas reality, a lot of it was dispersed and the like. Then a group from the University of Georgia came out with some information. CNN covered a group from South Florida, and then a group from Woods Hole came in. What does that say about that discussion about the difficulty in understanding the process of science?
It is difficult for lay people to understand the process of science. And one of the important things for us as researchers is to feel free to discuss our findings with our colleagues and with others without fear of being misinterpreted in the media or ridiculed. And it's been a very difficult working environment in the Gulf with that issue. Science is a process, and it's going to take a long time to answer these questions, and some of them are not going to play out for maybe a decade or so. And so we have to be patient, and we have to encourage that kind of science and research that will give us the answers that we need. But it's a process, and I think those two groups are going to have to work together to see who is right. And
right. Well, and the reality is they're not saying that the other one's wrong. They're just saying here's what we found.
Right.
And so over time it all kind of incrementally builds. But Jim, does that affect the work in your lab?
Well, I think Harriet pretty well described that. I think that's what we all deal with. We all understand the process. It's an in-depth, lengthy process. There's no reason to rush to judgment. And that happens, I think, in some instances, particularly during a crisis. That's understandable. But the bottom line is the scientific process usually works. It's a very effective process. It's a very tedious, at times, a very laborious process. It's a lengthy process. But in the long run, I think that's the best way to arrive at the proper conclusions, and the way we should all, it would help us all to better understand what really has happened, what really is happening, and the fact is we're going to have to continue doing that for some time to come, because this is, I believe, a rather long-term process, and we need all the answers we can possibly get. We all want to know what really happens.
Yeah. Well, do y'all have any frustrations about the unasked questions, or the questions that your labs can't get to at this point?
It's a very frustrating process. And we've touched on it before, when dealing with funding, because we have to continue to do those things that we are being funded to do, yet here is this opportunity and to work in additional areas, and yet the funding has not really reached those academic circles and research circles that it needs that they need.
Right.
Yeah, no, that's right. One of my colleagues the other day made an interesting statement, said there's so much that we need to do, so much that we have to do, but you know there are only 20 working hours in a day. So, but yes, we have many things, underway at this time. Of course, we're very interested in all of those, and we're doing the very best we can to answer these questions. And you know, we've been residents of the coast for 40, 50 years. We know a lot of people there. They know us. We know the community. We're constantly asked what's going on, what are you learning, what's the real story. You know, we want to know those answers as well, because we're part of that community as well. And so we and the Gulf Coast Research Laboratory faculty and staff, we're doing the very best job we can to get at these questions and to deliver answers as quickly as we can, but it does take a lot of time and it takes funding, much of which we don't have at this time.
Right. Yeah, that's one of the frustrations. I think GCRL is and its adjunct faculty, if you will, at the universities around the state are renowned in the United States for their knowledge of the Gulf of Mexico. Y'all were the first scientists to make comments when this was over. You were, because of your baseline data and all the research that y'all were actively doing. So yes. I think you're to be commended for that. Has there been any pressure put on you from government agencies to move into research areas where y'all have not been before? Do you expect that to happen? Not pressure. No, no. Well, we'd like to move into research areas that we see that need to be addressed that we haven't been able to because of maybe the lack of the sort of funding we'd like to have. That's pressure of one sort, I guess, knowing that there are things that need to be done, and you just can't get to them right now because of the inability to do that. Right. Well, I think that's sort of what I was talking about, what people call the unfunded mandates, where people start to expect the scientific community to answer questions for which there's no funding, nor there's been enough time to gather the information. And I think that's something that you all deal with probably on a daily basis with your research. It is. It's part of the way we do things, but now it's very critical. This is a crucial time, and we need a lot of answers to a lot of questions. And we're poised, and I think our laboratory is very well positioned to address many of these issues, and we're prepared to do so. Good. Well, what questions are y'all asking now that you think have the largest national impact, the largest northern Gulf of Mexico impact, and maybe the largest Mississippi impact? The questions that you're pursuing, how's that going to affect the audience?
I think one of the things that everyone wants to know is how will the dispersant and correxit affect the organisms that we're working on and actually the ecosystems themselves. And no one has ever, I mean, this is a new event to us, and it's very difficult for our laboratory in terms of the chemical analysis. It takes a lot of expensive instrumentation, so we're having to depend on our colleagues essentially to help us with all of those analytical tasks. And again, we keep coming back to funding. That's been a crucial issue with us. Right. Are y'all collaborating with the other labs around the Gulf Coast? Each state has at least one lab that's similar to yours in structure. Yes, and we probably need to ramp that up a bit as well, but I know Harriet, particularly in some of her work, is doing that. She's probably sort of leading the way at this point in those collaborations, I believe, with her specific work with crabs. Well, I know that I've spoken to a number of scientists, those other labs, and they always speak highly. Plus, they're all sort of interconnected by summer internships and graduate school and collegiality and research and that sort of thing. The issue on blue crabs, one of the frequently asked questions now is, did we learn anything from the Ixtoc oil spill in 1979 about blue crabs? Is there, I know the blue crab fishery is not as big in that part of the world as it is here, but were there any lessons learned that can be applied here? I am not aware of any particular studies on blue crabs from Ixtoc. But I think we will learn a great deal from this spill. There's been a lot of targeted research on blue crabs, so I think we're going to come up with some answers. Right. Well, certainly all along the coast, all of these commercial fisheries issues are just huge. They loom huge because of the economic webs that they represent. Webs. That not only food webs, economic webs, and then cultural webs about how everything's interconnected. And certainly that's playing out now with that seafood all over the country. And of course, people are very concerned about what the public relations side of that factor is. If people are going to look to the scientific community to answer questions about safety and about the future of these fisheries, and they're important to Americans. That's true. Yes. Yeah. Fisheries in the Northern Gulf are very interconnected. What happens in Louisiana is important to what happens in Mississippi.
Right.
Louisiana drives our fisheries both biologically and economically.
Yeah. Are there connections between the lab and the Extension Service, the State Extension Service?
Yes, our lab works very closely with an organization called the Gulf States Marine Fisheries Commission, in which scientists and managers and politicians from each Gulf state meet twice a year and discuss issues. And our next meeting will be in October, and I think it's going to be a very interesting meeting to talk with our colleagues in other states.
Well, good. Well, we have a few minutes left. Why don't we take a couple of minutes each, if y'all would sort of go back and hit the high points of what we just talked about and anything that you felt was left unsaid.
Again, I think as a fisheries biologist, of course, and having many years of experience on the coast, I've learned a few things over the years, and I think, of course, we're just extremely concerned about the future of our fisheries. We're hoping for the very best. But it's going to take some time, I think, for that really to play out, for us to understand what impacts occurred, the nature of those impacts, what the future of those impacts mean to fisheries on the coast, both commercial and recreational. And again, we'll be monitoring at a fairly high level on a variety of fronts to get at some of those answers and to try to better understand how things are working. How we proceed with our research in the future, and how we communicate to the public our findings and our continue to develop a good relationship and rapport with our citizenry along the Gulf Coast, because that's a very important part of this entire event.
Good. Harriet?
And I agree with Jim. I think Mississippi and actually all of the northern Gulf states are so fortunate in having a historical database that will allow us to really assess changes that we see. I mean, the database extends over a wide range of environmental conditions and climatic conditions. So we are really poised to be able to make those assessments. And assessment monitoring is a long, tedious, boring kind of science, but when you need it, it's critical. Yeah. And we have one of the best databases, I'd say, in the States.
Well, I agree with that, and I know that the media is just waiting on pins and needles to get information out of y'all's lab, and the kind of information that you're researching, because it's so vitally important to the well-being of people along the Gulf Coast. Yes. As well as our economy. So, thank you very much for being with us today. We appreciate your work, and good luck in what you have ahead of you. Aaron?
All right. Thank you so much, Dr. Thomas. And of course, we know that you have a lot of answers. So we want to let you know that you can find out more on our companion website at SpillScience.com. It's called SpillScience.com. There you can find in-depth information about topics related to the spill, as well as video discussions from scientists studying the effects of the spill in the Gulf of Mexico. You may also want to join the discussion on our blog section of the site, where you can engage in discussions with scientists studying the impacts of the BP Deepwater Horizon oil spill. Again, that site, SpillScience.com. And that's all for this edition of The Science of the Spill. Until next time, I'm Erin Pickens.
And I'm Bob Thomas.
This program was made possible by a grant from the National Science Foundation, a co-production by Mississippi Public Broadcasting and the University of Southern Mississippi Gulf Coast Research Laboratory.